Right. So number one, the subject of microbiology. Pretty much, you have to know the groups that I've listed here. Okay. These ones, and which branch of microbiology studies them, whether they unicellular or multicellular, and whether they have nucleus or don't have nucleus. Make sense? Understand why bacteria cannot form multicellular structures. Why? Huh? They can't communicate well. Okay. You may not see that exact answer, but you have to understand it's about communication, lack of inability to establish proper communication. Role and importance of microbes in the nature, industry, healthcare. Be able to give examples. Okay? Importance in nature, obviously it's everywhere. We talk about decomposition. We talk about I don't know, microbial sources of food for plankton in the ocean and stuff like that. In industry, we talked about fermentation, okay, food production, drug production. Healthcare, understand the idea of infectious disease. And understand which, I'm not going to ask you something very, very complicated, but you have to understand that, I don't know, injury, like a broken bone, is usually not infectious. Okay, things like that. While respiratory disease can absolutely be caused by the microbe, right? Classification. No 5 kingdoms. Can anyone tell me 5 kingdoms of life? Animals. Say again, animals? Fungi or fungi? Plants? Protists and Monera. Yes. Protists, Monera, fungi, plants, animals. Classification is based on what? What I'm saying is, how did he group them? Evolution, evolutionary relationship. What about those folks' classification? 3 domains, which are bacteria, archaea, and eukarya. 2 of the domains that are close to each other. Archaea and eukarya. Make sense? This classification is based on genetic similarities between specifically 16S ribosomal RNA. So when it says phylogenetic relationship, that's pretty much archaea and eukarya are closer. Okay? 2-word naming system for the microbes. I don't even want to touch on that. You understand? Genus first, species second. Okay. Milestones of microbiology. You won't have to list them. I will not ask you, list 10 important milestones. No. But you must recognize the roles for the scientists that you see in that table. Right? So it can be either direction. It can be a question, Edward Jenner, and you have 4 answers what he did. You have to recognize one. Or, this guy developed smallpox vaccination. Well, Edward Jenner. Does that make sense? I will try to make it, will do my best to make it as clear and differentiated as possible. Okay? I usually say it's 10 flashcards, and by the time you're done doing flashcards, you know them. You don't even need flashcards anymore. Structure of the water molecule, self-explanatory. It's a polar molecule that forms hydrogen bonds between partially positive hydrogens and partially negative oxygens. Implications are that hydrogen bonds are important for the structure of DNA, protein, carbohydrates, lipids, all biological molecules. Two strands of DNA are held together by hydrogen bonds. Good? pH scale. All you have to know, 7's neutral, above 7 basic, below 7 acidic. Period. You don't have to know the pH of Coca-Cola, which is 2.5, by the way. It's more acidic than your stomach. Macromolecules. I'm gonna talk about these four guys. So what's in common for each type? For nucleic acids, proteins, carbohydrates, and lipids. Well, lipids are separate. Know the structure. When I say know the structure, don't try to convince me that carbohydrates consist of amino acids. No building blocks, okay? For carbohydrates, monosaccharides make up polysaccharides. For proteins, amino acids. For nucleic acids, nucleotides. For lipids, it's slightly different. Triglycerides, glycerol and fatty acids. Phospholipids, phosphate, glycerol, and fatty acids. Function, know the function. For carbs, it's going to be energy and structure. For lipids, pretty much energy and structure. For proteins, mostly structure and regulation, enzymes. And for nucleic acids, storage and expression of genetic information. For lipids, phospholipids, cell membrane, no specific, okay? Triglycerides, energy. Phospholipids, cell membrane. Sterols and hopanoids, regulating the rigidity of the membrane, strength of the membrane. Sterols in the eukaryotes, hopanoids in the prokaryotes. Right? One thing that is common for all of them. How do we call the reaction in which building blocks make a long molecule? No. Dehydration. We take water away and we build a long molecule. When we add water, it's a hydrolysis, we break it down. Good? You got it? That's like those 4 slides. Okay? Genotype and phenotype. What is the genotype? All the genes that are there. What determines the phenotype? Which ones are expressed? Does that make sense? We all have very extremely similar genotype, but a different appearances because we have different gene expression. Does that make sense? Good. Now, second sentence here, I'm not really proud of it. Pretty much have an idea that expression of certain genes is determined by the environment. Not so much, question? So, determined by the environment. And expression changes as the response to changing environment. Okay? Evolution, basic mechanisms of evolution is genetic diversity, achieved in microbes via the exchange of genes and mutations. Underlying principles of microscopy. Refraction and reflection, physical processes. For light microscopy, know what magnification, resolution, and contrast are. Know how to calculate magnification. Know what limits the resolution. What limits the resolution? Frequency of light or wavelength. Okay? I will not ask you, and if I will, you will automatically get the point for that question regardless of how you answer just because. I will not ask you what limits frequency or wavelength. It's not the same, but kind of they talk about the same stuff. Does that make sense? So that's limitation. Major components of the microscope. All of them are pretty obvious. I want to remind you about two. The condenser that focuses the light on the specimen, and iris that regulates the amount of light that gets on the specimen. Everything else, I mean, objective lens, eyepiece, come on. Really, it's not rocket science, it's self-explanatory. Okay, major components, major types of light and electron microscopy and their application. The table, there's a table. Types of light microscopy, I don't remember, it's like 7 or 8 types. All you have to know is, I ask you this type of, you saw the questions. Against dark, not illuminated background, you pick dark field. This type of microscopy allows you to observe fluorescent image. Well, I will never ask you this because it's kind of obvious. It's fluorescent microscopy, right? I don't know. This type of microscopy uses lasers to observe samples in a thick slices of specimen, 2-photon microscopy. For electron transmission and scanning, know the difference. Yes? I may. And it will be very obvious. Okay? I guarantee it's going to be very, very obvious. You will definitely be able to tell, say, scanning from transmission. Okay? Does that make sense? Fixation. Why do we fix? So it doesn't move. Why do we stain? What do we increase when we stain? Contrast. Resolution. We can't increase resolution. We can only increase contrast or magnification. Major types of staining. Simple versus differential. In simple, you have to understand difference between positive and negative. Differential stainings that we talk about: gram, acid fast, endospore, flagellar, and capsule. Flagellar and capsule and endospore are self-explanatory. I will not ask you about the technique. Don't worry. I'm asking you about the type. Okay? I'm asking you about what it, based on what parameter of the cell you see the difference. So gram staining differentiates cells with different types of cell wall. Okay? Same goes for acid fast. Does that make sense? Before we go to the culture, any questions on microscopy? Okay. Microbial culturing steps. We start with inoculation, then incubation, then isolation, then identification, inspection and identification. You can put them all together. Inoculation. I understand the idea that you gotta collect with a sterile tool. It's great that we identified, that the confusion that my inoculation question caused. At least I know, so I will try to avoid this problem. Different types of media. So biological state, live and not live media. Okay. Physical state, liquid, solid, semi-solid. Chemical composition, defined versus complex. And finally, based on the function, know the difference between general purpose, enriched, selective, differential media. Does that make sense? Chris, question? No? I mean, you will see the question. Such and such media has such and such characteristics. Based on information that I provided, how would you call it? You have answers. Okay? Does that make sense? You have to pick one. I read through my questions several times. I do my best to make sure that there is no confusion there. I'm only human, and it's my questions. So I write them and I read them, so I often don't notice another point of view. Okay, so if you notice something during the exam, come and ask. Always, if you have a doubt, come over and talk to me. All right? Incubation. Why do we incubate in the incubator? Consistency, yes, to get reliable results, okay? So other people can reproduce them and we can compare results, between each other. Like I incubate my Monday class and you all, I can compare results. We can compare results between us because incubation happens in the same environment. Does that make sense? Great. Separation techniques: streak, pour, and spread plate. Pour plate is the synonym for loop dilution. Okay? Pretty much recognize the technique by description. Inspection. What can we see from inspection of the culture? Color. Size of the colony. Shape, appearance, How many, number, okay? For identification, it says describe at the basic level. What I mean by basic level, you don't have to give an extended explanation how we do Western blot, but you have to be able to recognize that method that identifies specific protein in the mixture of other proteins using antibodies to that protein is Western blot. Okay, protein method that recognizes, that identifies cells with a certain antigen using fluorescent antibodies is immunofluorescence. PCR identifies the unique fragment of DNA. Sequencing identifies the whole sequence of nucleotides. Okay, mass spectrometry, set of chemicals based on the mass to charge ratio. Chromatography, set of chemicals in the mixture based on the rate, how they go through the column. I will phrase it in a more English way, but okay. So you will have a question, either description of the method and the answers, and you have to pick one method, or name of the method and brief description, and you have to pick description that fits. Okay? Any questions on identification? Again, I do my best to make it clear, and you always welcome to ask me a question during the exam. I'm not kidding. I'm not teasing you. If it's something that you absolutely must know, like if you come and say, I have no idea what the word fastidious means, well, tough luck. Okay? If you come to me and I use some word and you don't know what it means, or if it's a term that we didn't cover yet, I will explain it to you right away. Make sense? Don't hesitate to ask me questions. If you don't understand the question, if you don't understand the answer, ask me. I will explain. Right? Because I don't want to hear after the exam, oh, I thought you were asking this. We have a little doubt. I'm right here. I'm not going anywhere. Okay. We've got this spontaneous generation. So, okay, this historical background. Well, we talked about Reddy and Pasteur experiments. Have an idea. what they are about. It kind of links to what we talked about, the major milestones. Know the contributions of Semmelweis, hand wash, Lister, surgical techniques, okay, aseptic surgical techniques, Koch, the postulates linking infection and disease, and Pasteur suggesting that microbes can be linked to the disease too. Koch actually first formulated the postulates. Endosymbiosis. Which two organelles are the result of endosymbiosis? Mitochondria and chloroplast. Clear? Why we pretty sure they are endosymbionts? They have DNA independent of the nuclear nucleoid DNA, and they undergo binary fission. They can replicate actually independently. Okay? Osmosis. 24. Major shapes and supracellular structures. So major shapes. Spherical. Name. Spherical. Coccus. Rod. Bacillus. Curved rod. Vibrio. Something between spherical and the rod. Coccobacillus. And spirochete and spirillum are self-explanatory. Spiral. Good. Supracellular. When I use that term, giving you a heads up, supracellular means above the cellular level. So when we see chain of cocci, it's streptococcus. When we see chain of bacilli, it's streptobacilli. When we see a bunch of cocci, it's a staphylococcus, like a bunch. And 2 cocci, diplococcus, 4 cocci, tetrad. That's that little table. Okay. So it's absolutely memorization part. Concept of osmosis, movement of water towards the higher concentration of solutes across the semi-permeable membrane. How it contributes to the cell shape. Understand what happens to the cell if it's in hypertonic or hypotonic environment. Now, what makes environment hypotonic or hypertonic? Where? Salts or chemicals? Where? Only? Yes. So if concentration, if osmolality in the cell and outside of the cell is the same, no matter how high it is, it's isotonic environment. Does that make sense? Now, implication of this, you have to be able to answer to the question how microbes can survive high osmolality outside of the cell. How? Yeah. Have high osmolality inside of the cell. Does that make sense? We talked about it. Alright? Major cell structures and their functions. What are the 4 cell structures that can be found in any bacterial cell? Nucleoid, plasma membrane, cytosol, cytoplasm, and ribosomes. 4. That's it. Everything else may or may not be there. Nevertheless, know the functions of them structures. Okay? Endospore, flagella, pili, fimbriae, know what they do. Inclusions. Again, I'm not going to ask you details that you don't know about. Okay? Routes for transport across the cell membrane. Active transport, what does it require? We'll get to that. Energy, ATP, okay. Does it go along the concentration gradient or against? Against. Diffusion, 2 types of diffusion. Facilitated, what does it need? Why we call it facilitated? Well, it needs a gradient. So it goes from high to low. What else does it need? Why it's called facilitated? Yes, some sort of a transport protein, transmembrane protein. Make sense? What is another type of diffusion that does not require transport protein? Well, it's essentially diffusion, but it's a kind of a just one case of diffusion. Passive diffusion, or simple diffusion, sorry, simple diffusion. It's passive transport, okay, that doesn't need energy. Simple diffusion, when chemicals move across the membrane directly. Which chemicals can do that based on the solubility? Fat soluble, right? Fat soluble can go across the membrane, water soluble cannot. We spent some time on the diffusion. Look at the slide. Cell walls, we discussed today. Know the structure. Membrane and thick protoglycan layer, peptidoglycan layer, sorry. Peptidoglycan layer in gram positive, membrane, peptidoglycan membrane in gram negative. Know what teichoic acid is. Know what LPS is. Know what the porin is and where you're going to find them or not. Know why acid-fast microbes are different. Okay, that they have mycolic acid on the outside. They're technically gram-positive, but with additional layer of mycolic acid. Archaea, just slightly different. The carbohydrates or glycoproteins. It's the part of the cell wall. Bacterial movement, know the idea of chemotaxis. Movement towards the increasing gradient, this time increasing gradient of attractant chemical. Yeah, so know things like capsule and slime layer and how they can contribute to pathogenesis and stuff. Any questions on that? Nope. Okay. That was pretty easy.